All right, in this lecture, we're going to talk about the Calvin cycle, which are the light-independent reactions. So these can continue when light is not present. They can also happen when light is present in most plants. We'll talk about some exceptions in the next lecture. But mostly the ATP and NADPH made in the light-dependent reactions is going to be used in these. So they don't depend on light, they depend on that stored up NADPH and ATP. This is going to happen in the stroma of the chloroplast. Remember, that's the fluid around the thylakoid membranes. Whereas, remember, the light-dependent reactions happen on the membrane of the thylakoid and in the lumen. And these reactions are going to fix carbon, which means that we're going to transform it from carbon dioxide, which is not a usable form, into a sugar, which is a form that can be used for energy storage and building a plant or building the photosynthetic organism. All right, so the Calvin cycle has three main parts. Phase 1 is carbon fixation, phase 2 is reduction, phase 3 is regeneration of the molecule that's going to start the carbon cycle. So just like the Krebs cycle, where we started with a 4-carbon molecule, in this one we're actually going to start with a 5-carbon molecule, do a bunch of reactions, and end up with that same 5-carbon molecule. Okay, so we start out with carbon dioxide, which is diffused into the cell and then into the chloroplast. And the carbon dioxide is going to bind to this molecule called rubisco. You may just write it like this or with the capitals like this. It doesn't matter. You do not need to know its full name, which is ribulose 1,5-bisphosphate carboxylase oxygenase, which is, it's just a bit much. Yeah. It means that there's a ribulose sugar with 2 phosphates, one on the 1 carbon, one on the 5 carbon, and it's going to interact with carbon dioxide. There, that's what its name means. Okay. Rubisco, which is what we will call it from now on, is going to bind a carbon dioxide molecule to this 5-carbon molecule RUBP. Ribulose bisphosphate, which has it's a 5-carbon molecule, bisphosphate meaning 2 phosphates, 1 on either end. Okay, so the rubisco binds the carbon dioxide to the RUBP, and then we end up with this 6-carbon molecule, which we split into 2 3-carbon molecules, 3 phosphoglycerate, and then we use the energy from an ATP to do that. Now, if you're wondering about, like, hang on, we have 3 of these and 6 of these, what? Okay, so we have to have 3 carbon dioxides go in to get 3 carbon molecule out. Except, wait, actually 6. So the numbers here are confusing. I'm going to try to keep them straight for you, but I don't need you to know all of those numbers. Okay, basically what I want you to know is carbon dioxide in, sugar out, and then where the energy for those things comes from, and rubisco, because rubisco is going to be our problematic friend. Got it? Okay. All right, so now that was our carbon fixation. We used the energy in ATP to split the 6-carbon sugar into 2 3-carbon molecules and phosphorylate them. Now, in reduction, we're going to turn this 3-carbon molecule into this 3-carbon molecule by taking a phosphate off and replacing it with hydrogens from the NADPH. Okay, we're reducing because we're also adding an electron. You remember adding an electron, adding a negative charge is reduction. So we fix the carbon into this molecule, then we reduce that molecule, and we end up with G3P, glyceraldehyde 3-phosphate, meaning that it is a glycerol, but aldehyde because it's got the hydrogens on it, and then 3-phosphate, meaning that the phosphate is on the 3rd carbon. This is the thing that is going to go out into the cytoplasm of the cell to get made into glucose and other sugars, such as fructose or galactose or any of those other sugars that the cell needs to make. It's going to make lots of those glucose molecules so that it can make cellulose to make itself bigger if it's a plant. And it's going to make starch to store energy as well. All right. So the G3P goes out of the chloroplast out to the cytoplasm, and then we get another G3P is going to go into this regeneration phase. Okay, how many of you are confused? Because this took me a really long time to figure out, okay? Because I just want it to be like the Krebs cycle, right? Where you get like the one pyruvate in and then just like it does it twice because you had two pyruvates that you made in glycolysis. But the Calvin cycle has to go a bunch of different times just to get to the regeneration phase. So one molecule of G3P goes to the cytoplasm. 5 molecules of G3P have to be used to regenerate the RUBP. What? Because we put 1 carbon on this 5 carbon molecule and split it into 2 3 carbon molecules. So we made 2, but we need a 5 carbon molecule back. It's confusing. Okay. So that is why why you need 3 CO2s to make 1 3 carbon molecule, but you actually use 6 ATPs and 6 NADPHs, because in order to do this whole thing, to regenerate the RUBP, you have to make 6 3 carbon molecules. Okay, do that 12 times, you get 2 G3Ps, and you can make a glucose. Wow, plants, so extra. Okay, so let's review everything that we've learned so far. Light excites electrons in photosystem 2 and photosystem 1. Photosystem 2 is going to use that energy in those electrons to oxidize 2 special molecules of chlorophyll, which pass electrons to an electron acceptor. They are reduced when water is split. Electrons go to the chlorophyll molecules, oxygen and hydrogen ions are produced. The electron acceptor, PQ, passes the electrons to the cytochrome complex, which passes the electrons to PC, which pass the electron to photosystem 2. In the meantime, PQ uses the energy from the electrons to pass hydrogen ions across the membrane so that we create a hydrogen concentration gradient. Yes. The electrons from photosystem 2 reduce the chlorophyll molecules in photosystem 1 that got oxidized because of the energy from light. Those electrons from photosystem 1 go to another electron carrier and are used to reduce NADP+ to NADPH. That NADPH goes to the Calvin cycle to power the reduction reactions here. The hydrogen concentration gradient is used to turn ATP synthase to make ATP. The ATP goes to the Calvin cycle so that its energy can be used to fix carbon and to regenerate RUBP. The Calvin cycle makes G3P. I always want to put the 3 first. And voila, we have fixed carbon dioxide into a usable form. We have broken water to produce a hydrogen concentration gradient that we use, and oxygen as a waste product. And that, my friends, is the light-dependent and light-independent reactions. Yay! And in the next section, we'll learn about other things that happen, because not everything always goes right.